Let's start the afternoon session. It is my pleasure to introduce Dr. Jason Yang as our Young Investigator lecturer today. So Jason is an assistant professor and chancellor scholar in the Department of Microbiology, Biochemistry, and Molecular Genetics at the Center for Emerging and Re-emerging Pathogens at the Rutgers, New Jersey Medical School. And his lab combines systems biology, synthetic biology, and machine learning tools to study mechanisms that drive chronic and infectious diseases, and focusing on antimicrobial resistance and host pathogen interactions. So Dr. Yang earned his PhD in biomedical engineering from University of Virginia, and he completed his postdoc studies at MIT and the Broad Institute. And his work is very well recognized with many honors, including the NIH Pathway to Independence Award, the Agilent Early Career Professor Award, and also our TSS 2025 Young Investigator Award right now. And his lab uses interdisciplinary approaches to advance models for disease prediction. So with that, let's welcome Jason.
Thank you. Can you guys hear me okay? Yes? No? Okay, I'm gonna stay on this side because that side of the room always gets all the fun. So I think the first thing I want to say is that it's a huge honor to be able to receive this Young Investigator Award, and so I'm deeply appreciative. And Sean gave a huge plug for ASM. And I just want to add a little bit to that, which is, this group, we're the branch here in New Jersey. And even though we're a small school, New Jersey, or a small group, New Jersey actually has a lot of schools in the state. And so, number one, thank you for the guys who drove like 2.5 hours from northern New Jersey just to get here. But the second thing is that, New Jersey is, we're a pretty great state, right? I mean, we do a lot of good things. We're a big part of the country economically. There's a lot of schools here, a lot of, people who go off and become scientists. So I think we should maybe give ourselves a little pat on the back and take some pride that we represent the state, and hopefully, in the coming years, we can get more of our friends to be involved. Okay, so I think what I'm going to do is so the title of my talk is about tuberculosis, but it's a little bit of a what's the word? It's a little bit of, forget the term. I'm a little bit of food coma right now, but I don't really want to talk about disease. I'm part of an infectious disease center. But really what I hope you guys can take away from this is a story about bacterial physiology and stress response, right? And so just, the overall premise of work in the lab, as Sam mentioned, is that we're systems biology labs. So we combine our experimental activities with mathematical modeling activities and machine learning. And the idea is that by bringing these tools together, we can do hypothesis-driven research a little faster. And hopefully that'll lead us to be able to translate some of our findings to do something useful and benefit mankind. Part of my lab focuses on tuberculosis. And for those of you guys who are not as familiar, tuberculosis is a global public health crisis. So it is the leading cause of death, of mortality and morbidity in the world, caused by infectious disease, by a single infectious pathogen. And that pathogen is Mycobacterium tuberculosis. And just for the purpose of giving a little bit of just context, to become familiar with this organism, because not everybody works with this, with TB. Mycobacterium tuberculosis is an acid-fast bacteria. Over evolutionary time, it's become actually a human-adapted pathogen. It is acid-fast, and one of the things that makes it pretty special is that, one of the things that makes it pretty special is that it has a really special cell wall. So unlike most bacteria that we study, like gram-positives have just like one cell membrane with like a peptidoglycan layer, or gram-negatives with 2 membranes, right? Mycobacteria have this really complicated waxy layer called mycolic acid layer, and one of the special properties is that these mycolic acids are really important. They do a lot of different things. One is they don't let anything get in, so it's really hard to get a lot of antibiotics or other molecules in. A second thing is that they are so strong and rigid that they play really important roles in just like in maintaining, kind of countering osmotic pressure, countering osmotic stress. So it turns out that this is a major, major target for most TB drugs. The second thing I want to introduce is antibiotics. So we all know what antibiotics are, but from a biological perspective, I think sometimes we forget what antibiotics do. So when I think about antibiotics, I think of them as small molecules, usually naturally occurring, whose targets are usually essential processes that are part of bacterial cell replication, right? So so there's a lot of really interesting physiology. Well, if a cell's not replicating, then a lot of times antibiotics don't work. But what I want to just say is that most antibiotics that are studied, most ones that we use in the clinic, target things that are directly connected to or part of central dogma. And because the target space is very narrow, right, and because of a lot of economic factors, it's really hard to develop new antibiotics. And so in recent years, there's actually been a new class of targets for kind of antibiotic drug discovery. And this has actually been specifically in bacterial metabolism, and even more specifically in processes that are related to generation of ATP. So there's a lot of new kind of antibiotic drug discovery programs that really target oxidative phosphorylation. And one of these, which we're going to be talking about today, is this drug called bedaquiline, which targets ATP synthase. Right? Okay, so a little bit about just antibiotics related to TB then, Mycobacterium tuberculosis. So it used to be, well, it still is, that if you come into a clinic with TB and you have, they do their laboratory tests and you're infected with a strain of TB that is drug susceptible, then the standard care is for a TB patient to receive this 4-drug regimen, which includes this drug isoniazid, rifampicin, ethambutol, and pyrazinamide, where 3 of these drugs target processes related to making the cell wall, right? In particular, today we're going to focus on isoniazid, and then rifampicin, as you guys may know, is a RNA polymerase inhibitor, right? So targets transcription, and so when patients come in and receive these regimen, most of them cure, about 5% don't. And then there are some that go off to develop drug resistance, right? And so, because these patients are seeing multiple drugs, oftentimes they'll develop multidrug resistance. And in the case of TB, multidrug resistance is specifically defined by being resistant to isoniazid and rifampicin. Make sense? And so in recent years, really just like in the last 2 or 3 years, there's been new drugs that have been approved to deal with multidrug-resistant TB. And the WHO just approved, I think literally 2 years ago, a new regimen that include bedaquiline, somewhere in here. That include bedaquiline for treating MDR-TB. And so this is some of like the biological context. So what I'm going to be telling you about today is a story that we published about a year ago, led by Nana Foriyaninam, who is a former postdoc in the lab and supported by other members of my lab that you see here. And so we're part of Infectious Disease Research Center. So of course, some of the things that my lab is interested in is trying to understand, okay, what are traits of drug resistance in TB? And because TB is such an important global problem, there are a lot of global efforts to try to characterize, like genetically, what is kind of the diversity of TB, what might be genetic traits associated with different kind of drug susceptibility phenotypes. And one of these was an effort led by what's called the CRYPTIC Consortium. I forget what that acronym stands for, but it was a WHO-supported effort to go and sequence TB isolates from around the world, and also measure the drug susceptibility profiles. And so they measure, I think in this study, there was something about 12,000 different strains of TB, and something like 12 MIC measurements or drug susceptibility measurements for 12 different antibiotics. And so we just became interested, and we're curiously just digging through the data. And one of the things that we noticed is that there was this interesting pattern where drug-resistant TB, multi-drug-resistant TB, had a lower MIC than non-multi-drug-resistant TB to this new drug called bedaquiline, right? These are population level kind of, measure, kind of like statistics. But there was a tendency for there to be greater sensitivity in multidrug-resistant TB to this new drug. And an important thing to qualify is that in both these cases, right, these are bacteria that are sensitive to this drug. So it's not as though one of these classes is actually resistant. They all will be susceptible to ATP synthase inhibition. They're all going to be sensitive to having some sort of drug disrupt their ability to make energy. But there's something special about the drug-resistant TB, Mycobacterium tuberculosis, right, that makes them even more sensitive to inhibition of their ability to make ATP synthase. And we dug around a little bit more in that data, and what we found is that the definitional MDR-TB is defined by being resistant to both isoniazid and rifampicin. And if we looked at those two components independently, this phenomena really seems to emerge from isoniazid resistance specifically. So in this project, we became interested in understanding, okay, why are these guys specifically sensitive to ATP synthase inhibition, right? So this is actually a physiology question, right? And so, just to validate this phenotype, because these were data that we got from some database, what we did was we did some experiments on TB clinical strains that we had available to us as part of our center. And these came from a collection that from like about 10 years ago. And what we did is we took TB clinical strains that were both drug susceptible or MDR we took MDR, we took just general drug susceptible, and we also took a isoniazid monoresistant TB clinical strain. And what you see here is that our drug susceptible and our dark black, which is H37RV, this is the laboratory model of TB that's most commonly used in TB research, right? That treated with the same amount of bedaquiline, the drug-resistant TB are much more sensitive. There's a significantly increased amount of killing, right? So we can validate that this phenotype exists. So one thing I should tell you about isoniazid is that when we deliver it as a drug, it's actually a prodrug, right? So when isoniazid itself is inactive, but when it gets internalized into Mycobacterium tuberculosis, it becomes activated by catalase. And so by the CatG protein, which is a catalase. And so for those of you who are not familiar, catalases are really important for bacteria to detoxify oxidative stress, right? And so what that means is that we reason that you can model isoniazid resistance, right, by using a catalase deficient mutant, right? So we received a CatG deletion mutant from some colleagues, and what we could show here in orange, right, is that the deletion mutant was hypersensitive to bedaquiline, right? So that means good. We have something that can reconstitute this phenotype that we're interested in, and importantly, right, if we complement it, the phenotype reverses. So at least we think that this is sufficient, right? And in addition, as I mentioned, catalase is important for activating isoniazid, and it turns out that in kind of the real world, catalase mutations account for something like 70% of isoniazid resistance amongst TB patients. And more specifically, of those 70%, something like 80 to 90% of those mutations in catalase occur specifically in like the S315 amino acid. So there's a very, very narrow specific variant of CatG that explains most of isoniazid resistance and is present in most MDR-TB cases. So we can validate our system both that if we delete this gene, these cells are basically not susceptible at all to isoniazid, right? And if we complement, we can both recover bedaquiline sensitivity and isoniazid sensitivity, or reversal of bedaquiline sensitivity. To validate this further, we worked with our collaborator Shuyi Ma, who's at the Seattle Children's Research Institute. And one of the things that's important to understand about CatG in terms of how it's regulated is that it's downstream of a promoter that can get repressed by FIR, which Jeff knows is one of the primary transcriptional regulators that bacteria use to respond to iron stress. And the reason for that is the cells accumulate too much iron. If you have a bunch of reactive oxygen species around, it's a really important source of generating DNA damage, right? So FIR is a inhibitor of CAT-G expression, and so what we reason is that if we overexpress, is that right? FIR is an inhibitor of CAT-G expression. So we overexpress FIR, then we're going to decrease the amount of CAT-G, and therefore we should also be able to reconstitute the phenotype as though we deleted CAT-G. And so that's what you see here. This is what we saw, if we delete CAT-G, and this is what they saw when they overexpressed FIR against like their empty control mutant, empty vector control. I told you that catalase is important for cells, just bacteria, to detoxify peroxide, detoxify oxidative stress. And so what we then did was we also just phenotyped and confirmed that our strains what they were by measuring susceptibility to peroxide. And again, what you see is that the deletion mutant is hypersensitive. We can complement that. And importantly, we can validate this in the clinical strains that we had. And so here in all of these remaining plots, what you're going to see is that the black plots, the black strains, are either on the left-hand side, our model laboratory strain H37RV, right, or on the right-hand side, drug-susceptible TB clinical strains. In orange, on the left-hand side, is going to be our CAT-G deletion mutant, right, our laboratory model of drug resistance. On the right-hand side, the orange is going to be MDR-TB. And in the cases where you see the red, it's going to be our mono-resistant TB. So these are cells that have, which we know actually has a mutation at S315 and CAT-G, but are not resistant to rifampicin. Okay, so we saw that these guys are hypersensitive peroxide. So one of the things that we wondered is, okay, is bedaquiline, is this ATP synthase inhibitor inducing oxidative stress? Is one of the ways in which the changes are caused by catalase deletion, explained by the inability for TB to handle any kind of oxidative stress being drained by bedaquiline. So this was, now this might be something that's a little bit counterintuitive, right? Because number one, oxidative stress is correlated with oxidative phosphorylation activity, right? And so if you inhibit ATP synthase, right, then your intuition might go to, okay, well, there shouldn't be more oxidative stress being generated. And the second thing was that a couple years ago, prior to our work, there was a paper published by a really big TB lab based out of Alabama and South Africa, which showed that if they did experiments with a series of different oxidative phosphorylation inhibitors, including bedaquiline doesn't actually generate any ROS in their experiments. And so we wanted to ask in our system, is bedaquiline generating ROS, and could this potentially explain what we see? So part of what we did, one thing that we did was to also kind of help us generate hypotheses. We performed RNA sequencing experiments on our wild type and our CAT-G deletion mutants with and without bedaquiline treatment. And so so we asked this question, is bedaquiline inducing oxidative stress? And so from our RNA sequencing data, the answer is partially yes, because we see induction of CAT-G, right, this oxidative stress detoxifying protein, which is part of like the general oxidative stress response that are common across bacteria. And also, in performing experiments with, sorry, the cursor's kind of acting funny. In these BSL-2 safe versions of TB, where, again, black is the wild type version, orange is the CAT-G deletion mutant version, where if we perform ELISAs if we perform cell ROX dye-based measurements of ROS, what we see is that in both cases, both the wild type and the CAT-G deletion version, that there is a significant increase in ROS being generated. This led us to reason that if there's more ROS, is the ROS potentially causing more damage inside of these cells? And so some of the ways that you can measure this is to perform ELISAs for protein carbonylation and oxidized deoxyguanosines, right? And so that's what we did in these BSL-2 safe versions. And in both cases, what you see is that not only is there more, does bedaquiline induce more, oxidative damage to proteins and DNA, but kind of the amount of damage that we see is even more heightened in these CAG deletion mutants in our model of drug-resistant TB. And again, maybe some of that's kind of intuitive because these guys have a really hard time dealing with oxidative stress. But remember, the insult here is you have these cells that are really directly responding to inhibition of the ability to make ATP. So we were looking at other things that were different in our expression data, right, our transcriptomic data. So one of the things that we then looked at was what might be differences between our wild-type cells and our CAG deletion mutants in the absence of stress, right? Because up until, before they see, because the way that we reason this is that their sensitivity to a drug will depend on whatever defenses exist at the moment that they see a drug, right? So in kind of like the steady state, untreated, unstressed condition. And so what we found was that when we looked at changes in gene expression, there were several transcription factor programs that were differentially expressed, right? And one of these is this RV3160C transcription factor. I forget specifically what's in its regulon. But so we hypothesized that perhaps what's happening is that by losing their catalase function, the cells are adapting physiologically by turning on other transcriptional programs. And that perhaps some of these transcriptional programs that they're turning on themselves may actually create some sort of vulnerability, some sort of physiological vulnerability to this kind of energy stress. And so what we did was, we took, through our collaborator at Seattle Children's Research Institute, we generated or received from them a, RV3160C overexpression mutant. And what you can see is that these overexpression mutants are also hypersensitive to bedaquiline. Both here in the middle, their ability to make ATP, as well as their ability to grow in the presence of drug. So one of the things that's really interesting is that this is work by Christina Stalling's lab, is that one of the genes that RV3160C regulates is RV3161C, which is actually, which also performs some redox stress, important redox stress functions, and which has been shown itself to regulate susceptibility or contribute to differences in drug susceptibility for isoniazid. Okay, so coming, based off of our, results where we saw increased oxidative DNA damage, we also wondered if perhaps there might be differences in DNA damage sensitivity. So then we looked to see in our expression data, were there differences in expression of, of DNA repair enzymes, right? Because we had more DNA damage in our resistant cells, especially on drug treatment. And so the short answer is yes. We actually saw a lot of changes, a lot of instances where the expression of different DNA repair enzymes was different between our wild type and our mutant cells in cases which were before they saw drug treatment. So that's going to be this first column and this third column, right? So these are untreated conditions. You can see there's lower expression of these DNA repair enzymes. And then following drug treatment, we see significant induction, right? So the cells are responding to DNA damage by turning on a very strong DNA damage response, where in their baseline, right, these defenses are much lower, right? So here are example of two different DNA repair enzymes. And so to validate this, what we did is we took our two strains, and we looked at their susceptibility to fleamycin, which is a DNA damaging agent. And what's replicated here in the clinical strains is basically yes. cells that have defective catalase activity or which are resistant to these drugs are also hypersensitive to DNA damage. Okay, so now I'm going to take I'm going to interject a little bit with a little bit of math and tell you about some of the systems biology work that we do. So one of the tools that's really, I think one of the things about science is that science is really fun. And science is really fun because you can bring together a bunch of different tools and kind of probe the elephant from different directions. And so as an engineer, one of the tools that we have in our toolbox are tools that help us study metabolism. And one of these tools are mathematical models that make predictions on the metabolic state of a cell based off of information like what's represented here, a map of all of the metabolic pathways that exist inside of a cell. So Sean was just talking earlier about Leringers, right? Or you can think of KEGG, right? Today we're kind of lucky that we have these giant maps of metabolism where we have, at a semi-comprehensive level, we know, from substrates all the way down to, to amino acids, nucleotides, the fundamental, components of cell membranes, the fundamental components that are needed to build a new cell. We have a map of what those reactions are and what enzymes mediate those reactions, or at least the ones that are enzymatic. And what you can do with these maps is you convert them into matrix form. And if you can convert them to matrix form, then you can do math sort of stuff. And so one of the ways in which these tools are really useful is that, well, you have gene expression data, right? You can say, hey, like these reactions in metabolism should be regulated by these enzymes. And for my gene expression data, I'll know whether or not that enzyme is there, right? And so if I have different RNA expression profiles, then I can use the model to predict what's happening in different conditions because the expression of different metabolic enzymes are going to be different. And so this is kind of like at a high level, how engineers perform metabolic modeling predictions from RNA expression data, and that's what we did. So remember, remember that bedaciline's primary target is in inhibiting ATP synthase, right? And we have these 4 different expression profiles now, right? So we have our wild type cells that are deficient in their ability to, express catalase, or cells that are unable to detoxify ROS, right? And cells, in both those cases, cells that are treated with this ATP synthase inhibitor. And so we were looking to see, okay, what might be some interesting places where the model would predict differences in metabolism. And there were actually a lot of places. So some of the things that we saw that I'm not showing you here are changes in metabolism that are relevant to nucleotide biosynthesis, which is something that I always found really interesting because that was something that I had spent a lot of time working on during my postdoc. But one of the really interesting things that we totally didn't expect were these differences in folate biosynthesis. And so for, just a reminder on biochemistry, so folate biosynthesis is really important for making these tetrahydrofolate precursors that get used by the nucleotide biosynthesis pathways. And some of the reasons why that's important is that it's also a really important drug target for antibiotics that we use to treat other kinds of infectious diseases, like trimethoprim, right? So, and cancer, and treating cancer. And so, what we saw was that, first, if you compare the first column versus the third column, that in the when the cells that are deficient in catalase that have a difficult time detoxifying ROS, they also have, in general, I mean, it's hard to see here, but they do have differences or defects in their ability to make folates, tetrahydrofolates. And now this gets amplified when these cells are experiencing ATP stress, right? So this is a model prediction based only from the RNA sequencing data. And so to validate this, what we decided to do was, well, we have our mutant strains and we have our clinical strains. Are these guys hypersensitive to inhibitors of folate metabolism? Because you would envision, you can imagine that if folate metabolism is important for cells to survive, right? And these guys already are defective in their ability to deal with, to produce folates under stress, then perhaps you can really amplify that, by using a folate biosynthesis inhibitor. So we did that. So we did that with both trimethoprim and a sulfonamide. And what you can see here is that, actually this is, yeah, so what you can see here in our clinical strains, which phenocopy what we saw in vitro, is that here in red are CACHI deletion monoresistant or CACHI defect mutant monoresistant clinical strain is hypersensitive to folate biosynthesis inhibition, but not the MDR-TB, right, which is really interesting. And so, this led us to hypothesize that perhaps the reason why we don't see this in MDR-TB is because we have that second mutation that's really important, right, which is this loss of function or defective function in RNA polymerase. Okay, so hold that thought. Okay, so I'm going to do a really, really short pivot to kind of a subsequent effort that we've been doing, which has been now studying TB clinical strains using some of these systems biology mathematical modeling approaches. So there was this really interesting study that was published about 10 years ago by the Norwegian Institute of Health, or whatever their Norwegian equivalent is of our NIH, where there was this patient that came in to one of their TB hospitals over a period of time, and that's what you're seeing here over these 4 years. This patient really, really suffered, right? But a patient came in and they received antibiotics over time, and those antibiotics really didn't cure that patient, right? And so as a consequence of receiving lots of different antibiotics, they developed really complicated forms of drug resistance. So first they developed multidrug resistance. So what this showing you here, the dotted lines, is when when the patient was coming on the visits, when they spit sputum onto an agar plate, and when colonies were isolated from those agar plates and sequenced, right? Versus kind of the treatment history of this patient. They eventually did cure, but it was after 4 years. On the bottom, so what they did was they did whole genome sequencing, of these different clinical isolates over time. And so what you see on the bottom is phylogeny. So what you're really seeing on the bottom is like this natural history of TB resistance evolution within a single patient. And in the paper, what they did was they decided to focus on these 4 time points. One in the beginning, when the patient first came in and they had drug susceptible TB, and in this instance, which is like a little over a year. Okay, well, just here SF4 MDR. That was the second time point, which was multi-drug resistant to isoniazid and rifampicin, and I think one of the other drugs. And then 2 later time points when they had developed what's called pre-XDR TB, which you can just think of as really, really bad drug-resistant TB. So they perform whole genome sequencing, but they also perform RNA sequencing on these clinical isolates. And so what Gautam Moretti, medical student in the lab, did was he also performed this genome scale metabolic modeling analyses on these different strains. And so what we're seeing here now is how metabolism is changing in TB as it's going from a drug susceptible state to a multi-drug resistant state to a really bad, pre-extensively drug resistant state. And so, without going into this a lot of detail, really what I'm trying to convey here is that metabolism is remodeling, as the cells are, acquiring each new layer of drug resistance. And one of the ones that some of the places that are really, really interesting, are not only processes related to cell walls, which are the target of isoniazid, right, but folate biosynthesis. And so the combination between, so the combination between, what we saw with our clinical strains, the difference between our multi-drug resistant and our single drug resistant, and these simulations that predict that folate biosynthesis is one of these pathways that gets altered as cells continue to acquire drug resistance, leads us to a hypothesis that we want to test, which is, is specifically this second mutation causing that reversal of the, of our folate hypersensitivity. Now, in the beginning of my title slide is talking about metabolic vulnerabilities in drug-resistant TB, and I kind of want to end on a translational note, right? So what I said in the beginning is that as a lab, we're really interested in understanding some of these mechanisms that explain some of these interesting phenotypes with the hopes that we can translate them. And so this example at looking at isoniazid resistance and bedaquiline is actually kind of a natural example of that, right? So isoniazid resistance is causing these changes. Acquisition of isoniazid resistance is causing changes in metabolism. And so now if you use a drug that hits metabolism, those guys become hypersensitive. The question is then, okay, well, if we, so the question is now, if we analyze these data from these extensive, pre-extensively drug-resistant TB, are there other metabolic vulnerabilities that are present there that can be targeted? And the short answer is yes. So there is one of the things that, one of the processes that popped up here, somewhere on this slide. Okay, well, actually, so yeah, so one of the things that popped up between the drug susceptible and MDR-TB were processes related to energy cofactor biosynthesis, so specifically menaquinones, which are some of those electron shuttlers, right, in oxidative phosphorylation. And so there was actually a paper published about 5 years ago where this group in Japan actually went bioprospecting for new antibiotic drugs, and they found a menaquinone biosynthesis inhibitor and found that it was these drug-resistant TB were hypersensitive to it. So this at least partially validates some of our predictions. Moreover, we looked at some of the other changes that occur, in the gene expression profiles. What we did was we then lined up the differentially expressed genes with data that's come out of, that's been published by Jeremy Rock's lab at Rockefeller University, who has done a lot of CRISPR-I screening. And we found that some of the transcriptional regulators that we found also elicit, drug sensitivity phenotype. So we think that kind of the combination of this metabolic modeling approach, as well as looking specifically at different transcriptional regulators, can help us identify new drug targets. And so just to summarize, so what I've shown you is that drug-resistant TB is hypersensitive to ATP synthase inhibition, and that the mechanism that underlies this really is caused by changes in the physiology of TB as it acquires new resistance-conferring mutations. And interestingly, we think that acquisition of rifampicin resistance actually corrects for or reverses some of the things that we saw in folate biosynthesis with under just isoniazid resistance. So with that, I just want to acknowledge here in blue the people who did the work. Nana Foriyanyam has now moved on to another lab, looking at host pathogen interactions. Here are currently the people, the really wonderful people I get to work with, and our funders. So thank you for your attention, and if we have time, happy to take any questions.
It's a really nice talk. So, as you pointed out, it's a little surprising in the sense that if you're inhibiting ATP synthase, you would expect the opposite. You would expect to have less reactive oxygen. When that drug was developed, presumably they found mutants that were resistant to the drug in terms of ATP synthesis. I mean, every drug ends up right.
Yeah.
So if you tested it in that context where you had a mutation with regards to ATP synthesis, is it possible you'd still see the effect? So maybe the BDQ is actually, so it inhibits ATP synthase, but the reactive oxygen is actually some off-target effect. Mm-hmm. And that could be a new target for, you'd want a drug targeting that.
Yeah, so let me answer that in a couple ways. So first, resistance mutations associated with bedaquiline resistance. So there's really two. The predominant one, that's the biggest one, is in MMPR5, which is a regulator of a transporter. So that's maybe not surprising, but that's off target. In terms of what's actually on ATP synthesis itself, it's on the epsilon subunit, so close to where bedaquiline is expected to bind, right? So where does a ROS come from? So one of the things that I didn't mention is that, so I told you that isoniazid gets activated by catalase. And the active form of isoniazid is actually, what catalase does is it takes NADH as another substrate and it modifies isoniazid to create this NAD, isoniazid adduct, right? So in that process, when catalase is active, it's also stealing NADH away from stealing NADH away from oxidative phosphorylation. The other interesting thing is that what we found in our separate work in E. coli is that when cells are under energetic stress, right? So in E. coli, what we've done is we've made a mutant that just burns ATP. It just futilely cycles ATP. When cells are feeling ATP stress, they try to make more ATP, right? So they start to try to push things further, they try to push more through oxidative phosphorylation. So they try to generate more NADH. It turns out that a lot of ROS that gets generated in a cell actually takes place at NADH dehydrogenase. There's like a series of iron sulfur kind of shuttling things that Jeff probably knows way better than I do, where superoxide is created. And so there's this combination. I think what's happening is that when bedaquiline is inhibiting ATP synthase, the cells are trying to push that system harder. And so now they're running that NADH dehydrogenase even more, and that's where the extra ROS is coming from. I'm going to get in trouble.
No, not at all. I was just curious if you ever tried to feed folic acid or what is it, PABA? I think that's an intermediate in folic biosynthesis.
That's another, yeah.
Try to kind of correct the phenotypes you see in the CAT-G mutant.
So we haven't done that yet. However, we have done an experiment like that, but I don't remember the results. So one of the
now you're in trouble.
So one of the things that we've done, in the lab is we've taken normal drug-susceptible TB, right? So I think part of the premise, one of the themes that I'm sharing with you is that metabolism, right, as a physiological aspect of bacteria, right, can directly or indirectly affect drug susceptibility. And so we have done experiments in laboratory HRS-NRB where we measure changes in isoniazid-MIC in response to systematically about 100 different metabolites. And so in there, folate's one of them. I think in our experiments, adding folate actually protects against isoniazid, but I can't tell you, if that right off the bat. And if that's the case, then it actually supports what I'm saying, but I don't really remember.
Quick question or comment, I guess. I saw that there's also purine biosynthesis or that's affected as well. Actually, the starting point, I believe, for the folate biosynthetic pathway is the GTP. I think it kind of diverges into different pathways eventually, so there might be that link as well, where if it's even starting earlier than that.
Yeah, so when I think about folate biosynthesis, so these are cycles, and so there's a lot of things that come in. I think about folate biosynthesis as starting with glycine and serine in one carbon. I don't remember where GTP might come in, but actually another fun fact that's related to, thank you for bringing this up. So there was a paper that was just published in Nature in June or July, which was published by Johnson & Johnson, who went off on a TB drug discovery program. And what they found was this new compound, JJ whatever, 4-digit number, that is a purine biosynthesis inhibitor, like a brand new drug. And basically they were showing, hey, purine biosynthesis inhibitor works. But what was really cool was in one of the figures they did an experiment which asked the question, is this new drug any better than some of the existing drugs that we use currently? And so the way they tested this was, right now, the way that the WHO approves use of bedaquiline for treating multidrug resistance is part of this 3-drug combination, right? There's bedaquiline, protalmanid, and linezolid. And linezolid, for those of you guys who don't know, is a protein translation inhibitor. And so what they did was they took they took TB cells, they treated it with BPAL, bedaquiline, protalmanid, linezolid, and they treated it with bedaquiline and protalmanid itself. And they treated with bedaquiline, protalmanid, and their drug, and the combination with their drug was the best. So, this might, support the idea that the addition of inhibitors against purine biosynthesis could also synergize with, be synergistically effective against drug-resistant TB. Yeah.
Jason, let's thank Jason again, and
thank you guys.